Okay. Where am I? There we go. All right, so let's talk about the urinary system. It's pretty much the kidneys and all the parts that come after the kidneys. Obviously, we won't get through everything, but let's get started. We'll get a good start on this. Hopefully, I answer a lot of questions, and hopefully you learn quite a bit from this today. So when we're talking about the urinary system, we're including, okay, the main organ is the kidneys, but we also are going to have some of the other structures involved, which are going to include the ureters, the urinary bladder, and the urethra. But we'll spend most of the time talking about the kidneys. So what do the kidneys do? They're a filter. That's essentially what they do. They filter the blood, and there's going to be some other roles that they play. So in doing so, when they filter the blood, okay, they're going to create this substance called filtrate. Eventually, as it moves its way through the kidney in certain areas of our kidneys, it will eventually turn into urine. So as a result of this process here, we find ourselves able to get rid of waste that have accumulated in our bodies. Okay. Also, we've learned in other chapters, both in this course and the previous course, that the kidneys help to regulate, all right, certain levels of ions in our body. For example, we talked about calcium quite a bit in the first part of this course in 210. And we talked about how calcium is very important for several things, for bone structure, for the operation of muscles, for the function of neurons. So it's important that we regulate its levels, and the kidneys play a big role in that. If you have too much calcium in your blood, then it can eliminate it through the kidneys into the urine. And then if there's not enough, it can prevent the elimination of calcium into the urine and retain it in the body. And it does this for all the important ions. You see sodium, potassium up there. We know that those are crucial in the proper functioning of our neurons. We'll learn later on about the importance of regulation of acids and bases. The normal pH of our blood is 7.35 to 7.45. So the set point, the middle range is 7.4. And so we want to maintain that proper pH. And sometimes our blood can become too acidic or it can become too basic. And so the kidneys play a role in balancing that out, and it'll alter the levels of hydrogen ions and bicarbonate. Hydrogen ions will act more in the role of acidity, and the bicarbonate acts more in the role of alkalinity. And so the kidneys play a huge role in that. Blood pressure. You know, a lot of folks think that the heart plays a big role, and it does play, you know, a significant role in the regulation of blood pressure, but your kidneys help maintain your blood pressure too. And then, of course, like I said, it's a filter. So the kidneys are going to get rid of, okay, molecules that are floating in our blood, hormones always, because they're traveling from point A to point B, all right, certain drugs that you ingest, antibiotics and whatnot. And so the kidneys will help in getting rid of those structures there. Right. It also plays a role in the formation of calcitriol. What's that? That's the active form of vitamin D. Vitamin D is crucial because it helps. Well, there's lots of roles for vitamin D with immune function, prevention of certain types of cancers. We're seeing more and more, but really in this course, we've talked about vitamin D in the resorption or the absorption of calcium from your gut, all right, into your blood helps to maintain calcium levels. Erythropoietin, okay, the kidneys produce that. We talked about that in the endocrine chapter, and the importance of erythropoietin when your blood oxygen levels become low. Okay, erythropoietin is produced, and that erythropoietin stimulates the red bone marrow to increase the number of red blood cells. And so as you know, the red blood cells, with red blood cells comes hemoglobin, and that increases the oxygen carrying capacity in our blood there. This is one of my more interesting facts in this chapter and in this course here, is because I just got done teaching one of my previous classes in 210 that the liver is important in the role of gluconeogenesis, and that's the formation of carbohydrates from non-carbohydrate sources like fatty acids, amino acids. And so that I've said in the past, that the liver is responsible for that, and that's true. Okay, but okay, the kidneys, all right, can actually engage in gluconeogenesis in extenuating circumstances. So just like in baseball, there's a backup catcher, a backup pitcher, all right. Well, our body has a backup organ that will help with the production of carbohydrates, glucose from those non-carbohydrate sources through gluconeogenesis there. So let's move in and talk about what filtrate is, and then we'll also learn a couple other terms, tubular fluid, and then urine. We all are pretty familiar with that. But filtrate is basically as the blood flows into the kidney, it's going to go through the structure called the glomerulus. And the glomerulus is going to filter out, all right, some of the real small substances, and that's going to include solutes, and it's going to filter out water from the blood plasma, and it is going to then start to move it through what we call our nephron system here. And so for the next couple of minutes, we're going to talk about that. We are going to talk about the path of filtrate as it travels through our kidney, through our nephron system there, and how certain substances, depending on what the needs of the body are, what our state of hydration or dehydration, our state of starvation or nutrient or rich state, all that's going to depend on what we are putting into the filtrate, and then eventually what we're going to be putting or taking out of the urine here. So again, that filtrate is formed as we move, all right, water and solutes and substances across the wall of those glomerular capillaries, that's our filtering system, all right, into this capsular space. And then when it's accumulating in that capsular space, that is then called the filtrate there. So now that filtrate is going to move into what we call the proximal convoluted tubule. Once it does, we give it a new name, and we call it tubular fluid. And we're going to call that substance tubular fluid, all right, for quite a bit as it's moving through our nephron system here. Okay, we go through the proximal convoluted tubule onto the nephron loop to the distal convoluted tubule, and then we move into the collecting tubules, and then eventually into our collecting ducts. And then it will remain tubular fluid until that fluid moves into, all right, our papillary duct. And that's going to be located within the renal papillae. And then that's when we officially call it urine. And so that's pretty much what you are going to be putting into the toilets, the urinal, whatever, all right, when you're going to the bathroom. That's that substance. We refer to that as urine. And so urine, once it's there in the papillary duct located in the renal papillae, okay, that will start to collect into what we call the minor calyx. And several minor calyces located throughout the kidney. And so the minor calyces will drain into the major calyx, and then there's a few major calyces, and then eventually all those major calyces will drain into the renal pelvis. And from there, the renal pelvis then is going to move the urine into the ureter, and then from the ureter, it moves into the urinary bladder, where it is stored until it's time for our micturition reflex to take over and eliminate urine from the urinary bladder. So this picture kind of shows you, I'll zoom in a little bit here. So here you can see there's our glomerulus, and then as we move from the blood, okay, through across the capillary walls here, into this space here, Bowman space, into that capsular space there, then we refer to it as filtrate. But once that filtrate then moves into our tubular system here, then we change names and call that the tubular fluid. So you can see it's tubular fluid for a while. It's going to go through all these various areas here, right, down into our collecting tubules, and then eventually moving down into the collecting duct. And then from the collecting duct, right, when it exits out of that into the papillary duct, right, then it will then be known as urine. And from there, right, our urine will collect into these smaller tube structures. Those are your minor calyces. And then those minor calyces will empty into our major calyces, these larger structures, right in through here. And then from there, all those major calyces will empty the urine into the renal pelvis. And then from there, from the renal pelvis down into the ureter and into the urinary bladder, and then out through the urethra. So that is the pathway of all of that filtered material from your blood, essentially from the kidney all the way to the urethra there. So now we're going to go into some detail all about how we are eventually going to form urine. So there are 3 processes that are involved in the formation of urine. It's very, 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 very important that you know the definition to these 3 terms: filtration, resorption, and secretion. So the first step, as we're going to start to make our urine, is going to be glomerular filtration. And that's essentially going to be what we talked about, that formation of that filtrate. So this is going to occur in the glomerular capillaries there. And so this is where water from your blood plasma, right, and things that were dissolved in the blood plasma, solutes, right, all sorts of different fun items there that are going to be filtered out from this area here. And so these solutes and the water are going to move into that capsular space, also known as Bowman's space there. And that's where it's going to start to accumulate. And then that is where it is called filtrate. And so how do we move from our blood into Bowman's space? Well, we're going to deal with pressure gradients once again. So the fluid and the substances that are dissolved in that fluid, the solutes there, are going to move from an area of higher pressure to lower pressure. So keep in mind, all right, we are dealing with pressure gradients, which are differences in pressure across some sort of membrane. And in this case, it'll be our filtration membrane, which we'll talk about here shortly. All right, the next term that you should know is resorption. Okay, so we're going to talk about tubular resorption. And so this is when, all right, our filtrate, that fluid now is moved into our tubular system. It is now called tubular fluid. And so we're going to talk about what's going to influence, all right, the movement of that tubular fluid, all right, across into or out of our tubular system. And you might be a little bit confused by that, but some substances might get resorbed back into our blood. Again, it depends on what is happening systemically. If you have a lot of calcium in the blood, too much, if you're what's called hypercalcemic, then most likely, all right, the calcium is not going to be resorbed and you will secrete it out through the urine. Okay, but now let's say you are in a state of dehydration. Well, we want to keep the water. We don't want to end up putting it into the urine. So what we'll see is the resorption of water. And that process, as you know, is known as osmosis, the movement of water from an area of high concentration to low concentration. So if you are in a dehydrated state, you want to retain the water. So it's less likely that you're going to be urinating it out. And so we'll see an increased resorption in water. So we'll talk about that. So the processes that are going to be involved in tubular resorption are going to be diffusion. You remember that all the way back from chapter 4, all right? The movement of a solute from an area of high concentration to low concentration. Then we have osmosis. That's the movement of water from an area of high concentration to low concentration. And then active transport. And there's a number of different types of active transport, but active transport, you should know, is going to require the use of ATP. Okay, diffusion and osmosis do not. Those are passive transport modalities here. So during tubular resorption, we're going to see materials that are in the lumen of those tubules, like the proximal convoluted tubule, the distal convoluted tubule, right? All the different tubules, and including the collecting ducts, right? We're going to see, right, those substances move from the lumen, and they're going to most likely return to our blood. So how do we do that? Through, okay, a couple different structures. You have what's known as the peritubular capillaries, and then you have another structure called the vasa recta. And so these capillary beds are going to be in close proximity to the tubular system. And so there's the location of where you're going to see the resorption of, all right, these substances. Now, obviously, anything that we require to keep us alive, anything that's vital, right, that will be resorbed. Unless we have too much or we're at toxic levels, we'll get rid of that, right? But for the most part, if you're just sitting and you're eating a relatively decent diet and drinking, a decent amount of water, right, pretty much all those vital solutes and most of the water that you consume will be resorbed. Right. And of course, if we have too much, then we'll leave that material into the tubular fluid, so it'll eventually make its way into our urine. And then our third term is tubular secretion. Right, tubular secretion, again, we're going to see here the movement of material, in this case, solutes. But in this case, we are going to be seeing the movement of these solutes through active transport. That's going to require some energy. All right, so this might, and I'll go over some of this maybe today if we have time. But if not, you might have to go back to chapter 4 and refresh your memories on some of the active transport processes here. Right, but what we'll see here with tubular secretion, right, that material, all right, is going to move, all right, from the blood, okay, back into our tubular system. So consider this: secretion material will move out of the blood, either in, all right, our vasa recta or those peritubular capillaries. They're going to move from those capillary beds out of the blood and back into our tubular fluid. And then eventually, okay, some of that material will be excreted into the urine. So these three terms, okay, filtration, think of, we're going from the blood into the tubular system, forming our filtrate. Then our tubular resorption is going to move those materials from the tubular system back into the blood. And then finally, tubular secretion, we're going to move, all right, materials out of the blood into the tubular fluid. And then again, depending on what's happening in the body and the systems there, will determine if that material that was secreted back, I shouldn't say back into that material, if it was secreted into the tubular fluid, whether or not, right, it will be excreted into the urine. So this is a great picture. This is figure 2410. I strongly urge folks to really pay close attention to this to get a good idea. Notice the color of the arrows and the color of the boxes above. They coincide. So that green box there, glomerular filtration, is showing you material that is in, okay, the capillaries here in your bloodstream is getting pushed out of the blood. And it's going to move here into the glomerular capsule in the space here. And that's our filtrate. And so that filtrate then flows into the proximal convoluted tubule. And then here in our tubular system, we're going to see at various places, notice there's arrows all over the place that looks bluish, purple to me, or I will say blue, right? They're all over our tubular system. And so they're going to move substances from the tubular system here, from our tubular fluid, into our blood. And then you'll notice tubular secretion, again, there's arrows all over the place. So that is going to move substances from the blood into the tubular fluid. So you could have substances that move into the tubular fluid here in the proximal convoluted tubule, and then they get pushed back into the blood right here. But later on down the road, they might get pushed back into the tubular fluid. So again, we'll talk about some of these and all this that goes on, but just notice, take a look at all the arrows, how they're all over the place, except for glomerular filtration that can only occur in one spot, right? But just don't confuse these terms, okay? Tubular resorption, right, from the tubular system into the blood. Tubular secretion is going to be the movement of those substances from the blood into the tubular system there. All right, so let's head all the way back to the beginning, to the glomerulus here, and talk about, all right, materials moving from your blood initially into that capsular space. And so it has to move across a membrane. That membrane, part of it is going to be the wall of the capillary. So we're going to talk about it. We consider this the filtration membrane. And so like any good membrane, right, we want it to be able to filter things out. So it needs to be porous. Also, it is going to be thin. And finally, it is going to be negatively charged. That's important, especially, okay, and if you didn't know this, that's okay, when we're dealing with proteins, because proteins are negatively charged, right? And so negatively charged things are going to repel from one another, just like positively charged things will repel from one another. Remember, opposites attract, like charges repel. So that's going to be important. All right, so what makes the filtration membrane? Okay, well, it's going to be made up of two structures, the glomerulus itself and the visceral layer of the glomerular capsule. Okay, the visceral layer of the glomerular capsule. So let's talk about these layers here. We're going to start the innermost. So you know that term endothelium, that is the wall of the capillaries in the glomerulus. And so there's three different types of capillaries, right? We have our continuous, we have our fenestrated, and we have our sinusoidal. Well, the fenestrated are going to be these capillaries that have these holes in them by design. So we can move things out of the blood and into that capsular space there. And so those fenestrations are pretty much going to allow the liquidy portion, so our plasma, the blood plasma, and it will allow dissolved substances in that blood plasma to pass through. So the endothelium really restricts a lot of the larger items, okay? Foreign blood elements, okay? Platelets, white blood cells, red blood cells, any of the large structures there that we want to keep in the blood, that's important. So those fenestrations will prevent, all right, the movement of large structures. All right, then, as you know, capillaries will have a basement membrane. So we have our basement membrane here. So along our basement membrane, we have our glycoproteins and our proteoglycans. This is a blast from the past here, all right? But these structures here, right, are going to be negatively charged. And that's important because, like I said, like charges repel. And proteins are negatively charged. So those glycoproteins and those proteoglycan molecules located in the basement membrane are going to prevent the movement of the large plasma proteins. Okay, some of the smaller ones might be able to squeak through, right? But we want to keep the large plasma, we want to keep proteins out of our urine, all right, if at all. If you see protein in your urine, right, then we start to have some concerns there. Finally, the third layer, right, of our filtration membrane is the visceral layer of the glomerular capsule here. And so this is going to be seen outside of our capillaries here. And so it's almost like a wrapping, and it's made up of these specialized cells, which we call podocytes. I'll show you a picture here in a second. And so the podocytes have these processes that come off the pedicels here. And so their job is, okay, again, to help with the filtration. And so they will surround the capillaries, but there's little spaces in between these pedicels here. And so these spaces create, all right, what we call filtration slits. And so it's these filtration slips that will also, in addition, prevent the passage of proteins, but in this case, the small proteins. So if they made it past the basement membrane, those small proteins, because they have a tendency to do that, well, then the visceral layer, all right, those podocytes there will prevent the movement, all right, of those proteins, those smaller proteins, right, into our filtration there. I can't not talk about the mesangial cells because we'll discuss their role here later on when we talk about some of the effects of hormones on the kidneys here. but we're going to see the mesangial cells are found within those various loops there of the glomerulus. And so they play a couple different roles here, okay? Phagocytosis, if anything gets out that shouldn't get out or gets stuck, all right, these cells will gobble them up, but they'll also play a role in contraction and change the ability of our glomerulus to filter out substances from the blood. Again, we'll talk more about this when we get into the blood pressure aspect of the kidneys, because the mesangial cells play a role in that. All right, so here's a nice picture of our filtration membrane. So if I zoom in a little bit, you'll notice here, okay, so here is the capillary, right? But you'll notice there's holes in the wall of the capillary. Those are those fenestrations. So again, that will allow liquid, all right, blood plasma to flow out, anything that's dissolved in the blood plasma. Some of the smaller proteins might be able to squeak out through these fenestrations here, but pretty much the large proteins and the large blood formed elements, they won't. They'll be trapped in the capillaries there. And then you can see right on top of the endothelium, there's our basement membrane. And so that basement membrane is nicely negatively charged, so it'll repel some of the negatively charged items that are flowing here inside the blood. And then the third and final layer is going to be the visceral layer with our podocytes here. And you can see here are other processes that are coming off, and those are the pedoceles. And so they create these filtration slits here. And those filtration slits, all right, will help to prevent the passage of those smaller proteins. Let's zoom in a little bit further here. So here you can see all of those, okay, all of those layers from a transverse view here. So here's our blood vessel here, and then here's the filtration space. So you can see, look, leukocyte, white blood cells, red blood cells, they're too big. They can't get out. If you're seeing red blood cells or white blood cells in your urine, you should be concerned because this glomerulus here, all right, this filtration membrane, there's something wrong with it. Okay, something bad is not, something not good is happening. So you want to check into that. Okay, but fluid will flow out. Okay, anything that's really small. Okay, so obviously liquid like water, okay, sugar like glucose, anything really small, ions, sodium, potassium, calcium, right? Some of our hormones will squeak through, right? Vitamins, anything that's really small, and our small proteins. Now, the large proteins, like I said, might not be able to make it through the filtration slit. And if they even do, well, this negatively charged, all right, structure right here will repel them and push them back. And if the smaller ones do manage to make it through, like this little fella here trying to get through, these filtration slits help to prevent the movement there. Okay, so how much of filtrate do we produce every day? A lot, a lot, 180 liters. Okay, if you want to get out your cell phone and convert that into gallons, be my guest, right? But it's a lot. These things are working, our kidneys, you have two of them, most of us do, right? They will be working 24/7. And so pretty much what we're going to see in our filtrate is going to be, all right, the blood plasma with this let me go back. Everything right here. This is what you're going to see. So pretty much the majority of, all right, the blood plasma, anything that's really small, anything that's been dissolved in the blood plasma, you will get some protein, but hopefully not a lot. And so this collects into the capillary space, and then from the capsular space, we move it into our proximal convoluted tubule there. And then we start to move that material through the tubular system. It's known as tubular fluid. All right, so what happens to some of the material if it gets caught in the filtered, the membrane here? Well, like I said, those mesangial cells will come through and they will gobble up any of that material and get rid of it, which is good, which is good. Okay, so we have three categories for the substances in our blood, right? We have the freely filtered, the non or not filtered, and the limited filtration. Okay, so in that green bubble that I circle here, all of that is going to be the freely filtered. Okay, the stuff that should not be filtered, like I said, are going to be the large item are the large ticket items, red blood cells, white blood cells, platelets, any of the foreign blood elements there, large proteins, right? They are not filtered. And then things that are limited in filtration are going to be some of the smaller proteins, proteins that are an intermediate size there. And some of the items, all right, are most of the things will be stopped from moving through because of those 3 layers there. Okay, so that brings us to our pressures. When we're talking about our glomerulus, we have to talk about, because I said we have items that move across this filtered membrane utilizing a pressure gradient. So here now we're going to talk about that pressure gradient there. So you've heard the term before, hydrostatic pressure. So in the glomerulus, okay, we give our hydrostatic pressure the term of the hydrostatic blood pressure referred to as the glomerular hydrostatic blood pressure. Then you can see the subscript of the G there. And so this is essentially, all right, the pressure inside of the glomerulus in those capillaries that are going to be pushing that material out of the capillaries across the filtration membrane there and into the capsular space there. And so obviously, if we have higher pressure in the glomerulus, right, then we are going to see movements of the materials from the blood into the capsular space. If it's a lower pressure, okay, we won't see that movement occur. So how do we increase the pressure in that area, or how do we decrease it? Well, that means we have to deal with the blood coming into the glomerulus and the blood exiting the glomerulus. And those 2 blood vessels are referred to as the afferent and the efferent arteriole. An easier way to remember which is which, the efferent arteriole begins with an E, so does exit. Exit starts with an E. Okay, so the afferent, AF, that is going to be the entrance arteriole into the glomerulus, and then exiting the glomerulus will be the efferent. So the afferent arteriole is larger. So that means, as you know, if you increase the diameter of a vessel, then more fluid can flow through it. And that can increase the pressure there. The smaller the diameter, okay, less flow, because we've increased the resistance, right, then we will decrease that pressure there. So blood pressure, or the glomerular hydrostatic blood pressure, right, is going to be the force that pushes things out of the glomerulus, while we also have, all right, pressures that are going to be working against that, right, that are going to oppose the hydrostatic pressure of the glomerulus. And so we have our blood colloid osmotic pressure. We should be familiar with that because we saw it before. And that is going to be, all right, that pressure, that pulling kind of force that is exerted, okay, by the substances that are in the blood. Proteins, for example, they do a wonderful job in doing this. And so they're going to create that osmotic pressure that is going to pull, almost like a pulling kind of thing, but it's that pressure that is going to keep items in the blood. So that's going to oppose filtration. So the hydrostatic glomerular pressure has to overcome that if we're going to move items out of the capillaries and into the glomerular space there. Right, so think of this pressure, the blood colloid osmotic pressure, as this pressure that's going to draw fluids back into the glomerulus. It's almost like a pulling. And then we have the capsular hydrostatic pressure. So this is going to be on the outside, on the other side of our filtration membrane there. And so this is that pressure that'll be in the glomerular space there, okay? And this comes from the filtrate. So again, that pressure can also impede the movement of fluid from the glomerulus into the capsular space, because it will exert a pressure pushing back towards the glomerulus, back towards your blood. So you didn't think you'd be able to get out of this that easy without having an equation So how do we figure out which direction, right, these fluids are going to move? So we have our net filtration pressure equation here. And so our net filtration equation is going to be, right, the glomerular hydrostatic pressure, right? We are going to subtract the colloid osmotic pressure, or the oncotic colloid osmotic pressure in the glomerulus. We're going to add that together with the hydrostatic pressure in the capsular space, because those are both going to be opposing our hydrostatic pressure in the glomerulus. So we add those up, take the difference. If, all right, we wind up with a positive number, that means then our filtrate will move from the glomerulus into the capsular space. So this example here is showing us 60 millimeters of mercury is our hydrostatic pressure in the glomerulus. Then we have our oncotic pressure of 32 millimeters. Then we add that to the 18 millimeter hydrostatic pressure of the capsular space. And that gives us a value of 50. We take the difference of these two values, which is 10. Positive 10 means that we are going to move substances from, all right, the glomerulus into the capsular space. If it's a negative, then we're not moving materials out of the glomerulus into the capsular space. And that's what this drawing here is showing. It's always good to have pictures. So here's our afferent arteriole, nice and big, meaning more blood, all right, coming in, more pressure occurring. So that will help to increase the hydrostatic pressure in the glomerulus. The efferent arteriole is much smaller. Okay, and then you can see, all right, from the values here, as we calculate everything out, we're moving, all right, our material from the blood vessels, from the capillaries into the proximal convoluted tubule. So our filtrate will be exiting out. All right, so what can affect, all right, our filtration pressure? Well, we have some variables there. So let's start off with this term called the glomerular filtration rate. Read the definition here. It's the rate at which the volume of filtrate is formed, right? We use the time that we use to calculate this is going to be 1 minute. So how much filtrate is formed, okay, in 1 minute? How much can that structure, right, produce? So if you see an increase in the net filtration pressure, then you have to assume you're going to see an increase in the glomerular filtration rate, because we're going to increase the volume of the filtrate that is getting pushed out of the glomerulus. All right. Other things that can increase the net filtration pressure are going to be, if we see more solutes that are being pushed out, think of solutes as an like a magnet, okay, that's going to draw water out of your blood. Remember, water it's a big follower. It just follows glucose, it follows sodium, it just likes to follow things. So if we increase our solutes, all right, then we will see more water moving out of our glomerulus and into the capsular space. All right, so another increase, if we see an increase in net filtration pressure, then we're also going to see more urine formed. And what we will see that's different, everything so far has been increase glomerular filtration rate, increase solids in water, all right, that are going to be in the tubular fluid, increase in urine, all right. We're also going to see a decrease in the resorption of the filtrate. Okay, so glomerular filtration rate, doctors use this quite a bit to assess kidney function. And it's a good measure, all right, to see what's going on, what type of physiological functions and conditions are occurring in the kidney, all right? And so it's great because glomerular filtration rate is tightly regulated. Your kidneys are always trying to keep, just like with your blood pressure, just like with your heart rates, trying to keep it in a certain zone. Okay, everything in that homeostasis, we want to maintain that healthy zone there. And so glomerular filtration rate is no different. It's going to be tightly regulated here. And so depending on what's, like I said, what's going on in your body, what are the other systems doing, what are the nutritional requirements that are going on, right? Hydration status is one example. Are you dehydrated? Are you hydrated? If you're hydrated, right, you have a lot of water, well, then that means you're going to move more of that water into the kidneys. And you've probably seen it when you drink more water, you have to pee more often. Okay, and so what we're going to see an increase in urine production because you have an increase, all right, in the water concentration in your blood. And so we want to keep that all tightly regulated there. All right, so what is going to influence the glomerular filtration rate? Okay, well, one of the items, variables that will influence that is how big is the lumen? Is it big or small? Okay, remember, the bigger the lumen of the afferent arteriole, okay, the more blood that can flow through, the more that we can filter. If it's small, you'll encounter more resistance, and there could be a problem with that. Another item that can influence glomerular filtration rate is, all right, our filtration membrane. Okay, if we increase or decrease the surface area of the filtration membrane, that will increase or decrease the glomerular filtration rate. If you decrease the surface area, okay, then we'll decrease the glomerular filtration rate and vice versa. Okay, so our kidneys, all right, are going to regulate our glomerular filtration rate. And so if the kidney all by itself, it regulates it, then we call that the intrinsic controls. And if something other than the kidney is going to influence or control the glomerular filtration rate, that's an extrinsic control. So I'll just start here with the intrinsic controls, and then we'll call it a night here. All right, so let's start off with what we call renal autoregulation, our intrinsic controls. And this is basically going to deal with the amount of blood pressure that occurs in the glomerulus. All right, now keep in mind that this autoregulation is going to be completely different than the systemic blood pressure or arterial pressure. Okay, so you can have fluctuations in your blood pressure. And the whole point is the intrinsic controls are going to try to make it where, even though you could have changes in blood pressure, all right, in your systemic circulatory system, your kidneys are going to try to not let that affect how they're filtering out your blood. So we have two mechanisms, myogenic response and then our tubular glomerular feedback mechanism. If you notice the term myogenic, M-Y-O, think muscles. Okay, so we're going to be dealing with muscles of some sort. We're not going to be dealing with cardiac muscle because that's located in the heart. And we're not going to be dealing with skeletal muscle. So we're going to be stuck with our smooth muscle. So the myogenic response is going to deal with smooth muscle located in our blood vessel. So the blood vessel that we're going to deal with is going to be the afferent arteriole. That's the entranceway that leads into the glomerulus. So the myogenic response is either going to be contraction of the smooth muscle or relaxation, but it depends on, all right, the amount of pressure. As you know, if we increase pressure, we increase stretch on the blood vessel wall. If we decrease pressure, we decrease the stretch. So the example listed here, we'll see, all right, we get a decreased blood pressure. So we decrease the stretch. So what we'll see is because of that decrease in the stretch, because there's less pressure going through that blood vessel there, then that's going to cause the smooth muscle cells in the walls of the afferent arteriole to relax. And as you know, this goes all the way back, all right, to when we were learning about the circulatory system, when the smooth muscle cells relax, then the blood vessels vasodilate. And as they vasodilate, they increase the size of the lumen, and that's going to allow more blood to enter into, all right, those capillaries, into the glomerulus. And so we'll see this occur if your blood pressure and your systemic blood pressure starts to drop. Remember, we want to always keep glomerular filtration rate constant. It needs to be maintained at a constant value. And so when your systemic blood pressure starts to drop, like for example, your hand gets chopped off, you start to bleed and lose blood, all right? A lot of changes are going on in your systemic blood pressure, but your kidneys are going to be undergoing this myogenic response. And so it's going to, again, try to keep that glomerular filtration rate normal. So, and then on the other side of this, right, if we get an increase in blood pressure, we're going to see more of a stretch on the vessel walls there of the afferent arteriole. And so what that will do is that will cause the smooth muscle cells to contract. And that contraction causes vasoconstriction. And so that will make the lumen smaller, which means less blood can enter into the capillaries of the glomerulus there. Again, we'll see this happen when our blood pressure increases. Okay, like when you're running, okay, and your blood pressure starts to go up, goes up to 140, 150, 160 over 90 millimeters of mercury. Right? But this will allow and maintain that normal glomerular filtration rate. So that's what we're seeing here in these pictures. They're showing us what's going on here. In the middle here, this is our normal systemic blood pressure. Everything's good. You're just sitting, actually, this is what's happening now. So you're sitting there listening to me. Okay, so blood's flowing through at a normal, nice, consistent, all right, rate of filtration, because your systemic blood pressure hasn't changed at all. Okay, someone bursts into the room, they cut off your leg, you start to lose, okay, copious amounts of blood, your blood pressure, your systemic blood pressure drops. That causes the vasodilation here of the afferent arteriole, trying to get more blood into, all right, the glomerulus here, again, to maintain that glomerular filtration rate. All right, week before all that happened to you, after you had your leg, before you got your leg chopped off, you went for a run. So you're out there for a run, you get an increase in your systemic blood pressure, and that increase in the systemic blood pressure, all right, is going to result in the afferent arteriole now undergoing vasoconstriction. And so it decreases that lumen size, less blood is flowing here. But again, we're trying to maintain, all right, that normal glomerular filtration rate. We're not letting that offset or change. And so that is the myogenic response. Okay, I'm going to stop here for this video.